Good morning, dear students. Today, our lecture is devoted to digestion and absorption of lipids in gastrointestinal tract. Last lecture, we discussed classification and structure of different groups of lipids, told that this group of substances is very different with many functions, but when we will talk about the metabolism, we need to say that this first stage is the digestion, and today we will talk mainly about this step of metabolism. But after absorption, there is a stage 2, intracellular metabolism of lipids, and here I want to give you overview to the different directions of catabolism and anabolism of lipids for the future, understanding that digestion is only first step of metabolism. So, catabolism of lipids, lipolysis, will include fatty acids beta oxidation in the mitochondria of the cells, oxidation of glycerol, of course. Anabolism of lipids will include synthesis of triacylglycerols and phospholipids that will be divided to the synthesis of fatty acids, elongation of them, desaturation, formation of different types of fatty acids. And then phosphorylation of glycerols and combination with fatty acids, phosphate for phospholipids, amino alcohols, and then. Very important part, this is the formation of lipoproteins for the transport, because we told that lipids are water insoluble, and to be transported they require attachment of well-soluble molecules like proteins. Synthesis of cholesterol for participation in the membranes, for bile acids formation, for the hormones synthesis and further, and ketone bodies formation. So look, many directions that we will discuss next lectures. Today we will be concentrated to the first step, digestion. Digestion of lipids is different from digestion of proteins and carbohydrates because nature, properties of lipids, their hydrophobicity plays great role in the digestion mechanisms which necessary for, especially for such hydrophobic molecules. Let's do the overview to the step of lipids digestion with the help of such a good man. So we are eating different lipids, but the most important sources for fatty acids and glycerol for us and cholesterol, of course, are triacylglycerols of our food, cholesterol esters, and phospholipids. The first step, this is the quick interaction of dietary lipids with enzymes from oral cavity and stomach. A little bit further we will talk in detail. At this moment we are talking that step 1, this is the interaction with lingual lipase and gastric lipase. More longer and more complicated step 2, this is a interaction of lipids with the pancreatic lipase, cholesterol esterase, and different phospholipases that are synthesized and secreted to the small intestine by pancreatic gland. Step 3, which is combined with the step 2, this is the secretion of bile acids. Together with the bile, liver is secreting bile acids, very necessary for the correct digestion and absorption of lipids. And step 4, this is the formation of mixed micelles and absorption of lipids. Mechanism is very difficult for the absorptions. And fifth step, last step, this is the formation of lipoprotein particles of the lowest density, which are named chylomicrons, and they travel through the lymphatic system to the blood to be transported then to different cells of our body. So this is overview. We are dividing digestion and absorption of lipids to the different steps, and we'll talk about them more detailable one by one, step by step. So the first, in oral cavity, we have enzyme which is named lingual lipase. Lingual lipase is synthesized by the special cells that are situated in the tongue, mainly in the back of tongue, and in the saliva this enzyme is present in small quantities, but really in adults it is not significantly influent to the digestion of lipids. Why? At first, food is not very long time we are keeping in oral cavity. The second reason, that pH optimum for this enzyme is a little bit acidic, near 4.5, 5.0. You know that the pH of saliva is a little bit more alkaline, so this enzyme will not be in optimal conditions. Due to this, in adults, there is no significant activity of lingual lipase, and its participation in the lipids digestion is not so important. It is important for infants, for neonates, because the pH of their saliva is a little bit another, more acidic. Lingual lipase is a representative of acidic lipase, so called, in comparison with alkaline lipase, which is pancreatic. Moreover, this enzyme specified for the cutting from the triacylglycerol fatty acid with short and medium lengths of chain. Especially in the breast milk, such fats are present, and you know that the milk will be is keeping by babies in oral cavity a little bit more longer. So this enzyme is specified for the milk fats, and these short and medium chain fatty acids are very important source of energy for the babies. So we made the conclusion that in oral cavity lipolysis is started, but with not very high velocity, because lingual lipase has no equivalent optimum of pH, and the time for the keeping of food in the oral cavity is not so long in adults. Next step, this is the stomach, where the first mechanical emulsification is beginning. We need to say what is emulsification and for what it is necessary. Our enzymes are proteins, they are water soluble, and they can act only in the water medium. They can interact with the fats as a substrates only on the surface of lipid drop, because here we have the interface between lipid and water. So to increase the square of this surface, it is necessary to cut this big drop to the small drops to increase the possibilities for the enzymes to come and to interact with fats which are necessary to digest. So emulsification is a very important step for lipids digestion, and it can be mechanical. You also, I hope that you all understand about what I'm talking. When you are eating fat soup, for example, you can take the spoon and cut the big spot of lipids on the surface to the small drops. But if you will stop to do this, all the drops will glue again together to the big spot, because mechanical emulsification is not supported by anything. So you will cut the drops and they will glue together again if there is no something that can cover them and act against of the gluing of them again. But mechanical emulsification also important, because in the stomach we have enzyme which is named gastric lipase, many lipases we have in the body. So let's start to recognize them and differentiate them one from another. Gastric lipase, produced by the cells of stomach, but also it is acidic lipase, but you can see that optimum of its action between 3 and 6 also is not equivalent to the pH which is really exists in stomach of adult person. I hope that you remember in the stomach we have very important protease pepsin, which is necessary for digestion of proteins, and these enzymes optimum of pH is very acidic, near 1.5, 2.0. So especially for this, the great quantities of hydrochloric acid is synthesized together with the gastric juice to create this optimum of pH. So gastric lipase in adult people also is not working in the optimal condition. It is working, but velocity is not very high. Anyway, part of lipids can be digested even in stomach. The importance of these enzymes, same like lingual lipase, is determined by its action in infants, where pH of stomach is more alkaline, near 4.5. Milk fats also can be significantly digested in the stomach of babies due to the presence and due to the higher activity of gastric lipase in neonates than in adults. To come further, we need to see the process of emulsification with the help of special stabilizers. I told you that mechanical is not stable mechanism of emulsification. For the action of pancreatic enzymes, we need something that will stabilize our small drops of fats, and these are bile salts. About structure we will talk a little bit further. Here there is a scheme how bile salts will surround and stabilize small drops that are suspended in the water solution in intestinal juice, in the chymus. It is necessary to increase the surface of fat for the enzymes, as I told you, and if lingual and gastric lipase do not require bile salts for the stabilization of emulsion, pancreatic enzymes require this, and especially for these enzymes, bile salts are necessary. So bile must be present in this lumen of intestinum to help digest lipids. So, what is the function of bile acids? These are a list of enzymes that mainly are acting in the first part of small intestinum, duodenum, pancreatic lipase, phospholipases, cholesteroliesterase. These enzymes are synthesized and secreted by pancreas. Pancreatic lipase. Name is based on the place of production, so don't confuse with any other lipases which we have in our body. Bile acids are acting both to the fats and cause their emulsification and stabilize the emulsion of the fats, and to the inactive lipase. All pancreatic enzymes, especially proteases and lipase, are secreted by pancreas in inactive state to prevent outer digestion of the gland. Lipase is also active. Not only proteases are dangerous in the active state, lipase is also very active and aggressive enzyme if it is activated in the lumen of pancreatic duct or will be activated in the blood. It's a very difficult situation for the treatment. So pancreatitis is a very severe problem of medicine. In healthy people, activation of lipase will be in lumen of duodenum only, and bile acids are necessary for this process also. So, from the inactive state, with the help of bile acids and one more amphipetic peptide colipase, we will talk about it a little bit further, lipase will be activated and act to the emulsion of fats that will be cleaved after this. So at this moment we can write the most important functions of bile acids. First, emulsification of fats. Second one, activation of pancreatic lipase. You can write the third one and fourth one, that bile acids helps in the maintenance of pH in the duodenum, necessary for the activity of pancreatic enzymes. Bile acids, or bile must be sterile, and bile acids also helps in the anti-infection protection. So they will also next as anti-microbic agents. And the next function, this will be participation in absorption. In detail we will talk later, but it is possible to write now that bile acids also participate in absorption. So emulsification, activation of lipase, pH maintenance, antimicrobes, and absorption of lipids. These are main functions of fatty acids, of bile acids, sorry. Again, emulsification increase the square of the surface of the droplet, especially to give the ability to give triacylglycerols, most important sources, dietary sources of energy for us, for the action of lipase. For this we require bile acids or bile salts of them, which are amphipetic. They have charged part of molecule that can interact with the water, and hydrophobic part of molecule that can interact with the fats. And to represent this hydrophobic core of the small droplets, colipase is necessary, which is secreted together with lipase and activated by trypsin here. Colipase is helper for this mechanism. Look to the structure of bile acid and bile salt. Bile acids are synthesized from cholesterol. You can see this cyclopentan perhydrophene and trannic ring, characteristic for all steroids. So bile acids and bile salts, these are representative of the group which is named steroids, yes, together with cholesterol. They are synthesized from cholesterol in liver, and you can see that here they are acids, so they have a carboxylic group, which is ionized, of course, in the normal water solution. So we have two parts in these molecules. They are hydrophobic part, which is represented by this part of cholesterol, by these rings, which are hydrophobic, and more hydrophilic part, where we can see the carboxylic group. Hydrophility of this part can be increased by conjugation of bile acids with different water-soluble molecules, like amino acids, like taurine, which is also synthesized from amino acid. And here you can see the conjugated bile acid. If it is interacting with the sodium, for example, ions, we will talk about bile salts. So it's not a mistake to name these molecules bile acids or bile salts. It depends from their state. Here you can see the glycocholic acid, which where the cholic acid is conjugated with the glycine, with small amino acid, so appearance of additional nitrogen atom will increase the hydrophility of this part. So this hydrophilic head will look to the water, hydrophobic part will interact with the fats in these droplets, and by this way salts, bile acids will stabilize the emulsion and gives the possibility for lipase to digest triacylglycerols. Under the action of pancreatic lipase, triacylglycerols, let's remind yourself the structure: glycerol, 3 residues of fatty acids in 1, 2, 3 position. With the help of pancreatic lipase, 2 ester bond, mainly from the outside of the triacylglycerols in the position 1 and position 2, can be cutted, and we will obtain this substance that is named 2-monoacylglycerols. So monoacylglycerols without indication of the position 2. Look, interesting that in monoacylglycerols they are a little bit more water soluble, of course, than triacylglycerols, because here we had 3 3 tails of fatty acids which are absolutely hydrophobic. Here we have only 1, and 2 alcoholic group of glycerols instead here. So also such a molecule can be oriented in the space with a little bit more hydrophility from the side of alcoholic group, and a little bit more hydrophobicity from the side where the tail of one fatty acid is present. Usually this third fatty acid is not cut from the triacylglycerols. It can be done even spontaneously under the action of the different substances from the chymus or mechanically, but mainly absorption of the remnant of triacylglycerols will be done by absorption of free fatty acids and absorption of monoacylglycerols. So look, this is the mechanism how pancreatic lipase will cut free fatty acids from triacylglycerols. Products will be free fatty acids and monoacylglycerol. This is necessary to write because very often you are talking about degradation of dietary lipids till the free fatty acids and glycerol. As I told, very small quantity really is degraded till glycerol, free glycerol. Most of the triacylglycerols in the intestinum and our duodenum will give monoacylglycerol. But it will be also good because monoacylglycerols will help for the absorption, for the formation of the particles and further absorption of lipids. In addition to the pancreatic lipase, pancreas is secreted phospholipase. Phospholipases are necessary for the cleavage of phospholipids. Phospholipids differ from the triacylglycerols by presence of residual phosphate and something that is combined here. It can be amino alcohol, it can be the residue of monosaccharide. So we have different phospholipases which are specific for the special bonds that we have in phospholipids. To cut residue of fatty acid in the position 1, we have phospholipase A1. To cut the second one, phospholipase A2. Possibly for us to cut phosphate and amino alcohol or residue of monosaccharide in this position, but for this we need the special phospholipases which are named phospholipase C and D. For us, of course, very important significance, the phospholipase A2 that are present inside of our cells, not in the lumen of intestinum. It is also named phospholipase A. But here, the phospholipases of pancreas and acting in our intestinum are represented. So phospholipids can also degrade in our intestinum and gives us products of this reaction, free fatty acids, phosphate and amino alcohols that after this can be absorbed also. So we are, one word more, esters of cholesterols, if they are coming, also will be deesterified, so free fatty acids will be cutted from the alcoholic group of cholesterol, and absorption, for the absorption we will take from cholesterol esters free fatty acid and cholesterol. Cholesterol ring is not degraded in the lumen of intestinal. Moreover, cholesterol ring is not degraded never in any, no degraded completely in our body. We will excrete this ring with the help of our gastrointestinal tract and bile acids. Anyway, cleavage of ring of cholesterol will not be done in intestine. In small intestine, the process of absorption is started in lower part of small intestine. Put attention that part of resulting products after digestion, after cleavage of lipids, is water soluble. So if small quantity of free glycerol is formed, it can be absorbed easily to the blood because glycerol is a water soluble molecule. Also fatty acids with short chain, with number of carbon less than 10, can pass through the intestinal wall without any help by themselves because they are water soluble. But more longer fatty acids with the number of carbon more than 10, together with monoacylglycerine and cholesterol, must form mixed micelles, and only after this, these mixed micelles can be absorbed to the enterocytes. So when we talk about absorption of amino acids or monosaccharides, everything was easy. The special carrier proteins that will transport from the lumen of intestine to the enterocytes and then to the blood. No problem. Here we have a problem because fats are fat soluble and it is necessary to put them from the water chymes to the water medium inside of enterocytes at first. Then we will have the next problem, how to put them to the blood or to the lymph. But at this moment, to cross membranes of enterocytes, the hydrophobic long-chain fatty acids, monoacylglycerines and cholesterol must form micelles. What are micelles? These are particles of orientate, especially orientate lipids. As we told, in our lipids, in our products of degradation, free fatty acids, monoacylglycerols, and present already bile salts, bile acids, there are hydrophobic and hydrophilic part of molecule. So if these molecules will interact one to another, look, with their hydrophobic parts, free fatty acids, look, yes, free fatty acids, their carboxylic group is hydrophilic if ionized, they can interact one to another with their hydrophobic tails and glue by them. Monoacylglycerols also can be oriented. Hydrophilic alcoholic group of monoacylglycerols will look outside. The tail of residue of one fatty acid in the second position also can glue by its hydrophobic tail to other fatty acids and to other monoacylglycerols. Finally, bile salts with their hydrophobic ring part will interact inside with the hydrophobic tails of fatty acids, and its carboxylic group, which is ionized, or maybe a residue of glycine or taurine, will look outside. So cholesterol, free cholesterol also can be incorporated between these substances because its alcoholic group can interact with water, yes, and in this view, this mixed micelle will be absorbed to the enterocytes. So this is the last function of bile acids, to participate in absorption of lipids. Put attention to the structure of mixed micelle. As I told, in bile salts, hydrophobic part interact with the lipids, with the fatty acids, and then gives ability to the lipase to cut, and free fatty acid and monoacylglycerols after this that are formed helps in the formation of this particle. Finally, starting from the big fat drops, with the help of gastric lipase, first free fatty acids are formed and help to stabilize this drop. Action of gastric lipase can do and mechanical emulsification can do these drops a little bit less, and when they will come to the lumen of duodenum, pancreatic lipase will bind with the colipase and bile salts to start digestion of triacylglycerols till free fatty acids and monoacylglycerols. Look, they are shown here. Also, look, diacylglycerols helps to stabilize the drops, and finally micelles are formed with the help of free fatty acid, with the help of monoacylglycerols and small quantity of diacylglycerols. And micelles go diffuse through the brush of the enterocytes and then absorbed through the brush of our enterocytes inside of them. So, big droplets that are surrounded by the monoacylglycerols, phospholipids that are present in the bile also helps to stabilize these structures, go through the gastrointestinal tract, become less and less emulsified, digested, and then resulting products, mainly free fatty acid and monoacylglycerols, are absorbed with the structure of micelles inside of enterocytes. Then the next stage is beginning. This is overview on this stage. Inside of enterocytes, there will be a resynthesis of the triacylglycerols. Why it is necessary? We are eating fats of different biological species, from plants, from animals, and they have the species-specific content of fatty acids in their triacylglycerols. But inside of our body, we require in the triacylglycerols which are specific for human body. And due to this, at this first stage, we are resynthesizing triacylglycerols for their further transport in the particles that are named chylomicrons. This will be done inside of enterocytes. So the first big site of synthesis of our fats, this is the mucosa of our intestine. We are resynthesis of triacylglycerols, resynthesis, esterification of cholesterol will be done, and then during this process, more specific for us fatty acids will be incorporated into the structure of fats that after this will be transported. Chylomicrons after this will go at first to the lymphatic vessels, and lymph will be collected from the all of the body, and you know that big lymphatic duct will be combined with our venous system. Finally, chylomicrons, with the help of lymph, come to the blood. There was an experiment. Only if quantity of fats in the diet is very big of the one species, it is possible to find in the adipose tissue of the organism these non-specific for this species fats. For example, there was experiment when the dog was fed with the seed oils, and finally scientists observed change of type of fats in the adipocytes of this dog to the more similar to the seed oil. But really, all the fatty acids that we are eating, we will use, but not for the construction of our triacylglycerols. When we will construct our triacylglycerols, of course, we will incorporate fatty acids which are specific for human. And our fats differ from the fats of chicken, of lamb, of pigs or cows, differ from the oils of seeds and grains. We will synthesize our fats. So this moment we will talk a little bit later more detailable, because at first I want to put your attention for the disorder of absorption of lipids, which is named steatorrhea. Steatorrhea, it's a excretion with the feces big quantities of fats. They can be visible under microscope, but moreover, patient can see presence of fats in the feces by the formation after defecation on the bowl of toilet. The special stain, spot or stain with fat gloss, and have a pain in the top of abdomen, have a different problems with the defecation, so-called oily defecation. Everything of this science, this is about steatorrhea. Right, please. What is this? You have this question even in exam questions. Disorder of lipid metabolism, and the first is a disorder of the digestion and absorption. What reason can lead to the steatorrhea? At first, alimentary nutritional reasons, when a person consumes so big quantity of lipids with the diet, and even healthy organism cannot digest so big quantity. Of course, excretion of these lipids will be visible, and treatment in this case is not necessary. It is necessary to normalize the presence of fats in the diet. Second group of reasons, this is the problems with the secretion or excretion of bile. If it is irregular or bile has a changed structure, or if there are stones in gallbladder, in this case, hepatogenic steatorrhea can be observed. So in this case, it is necessary to normalize excretion and secretion of bile, and steatorrhea will be treated. Next group of reason, it's so-called pancreatogenic steatorrhea, and understandable that excretion of big quantity of fats with feces is possible if they are not digested and not absorbed after this. For absorption, digestion is necessary. So if pancreatic gland is not secreting enough quantities of enzymes, or there are no conditions for their activation in the lumen of small intestine, pancreatogenic steatorrhea can be observed. And finally, enterogenic steatorrhea, where the problem is with absorption of lipids. Maybe this is inflammation of the mucosa of intestine. Maybe this is inherited problems with the membranes of enterocytes. All of these problems will cause no absorption, and lipids will be excreted with the feces. So all of these signs, compliance of the persons with steatorrhea must be recognized by the physician, and check of all of these mechanisms will help to find the reason for steatorrhea and choose the method of treatment in this each case personally, individually. Let's return to the enterocytes, where absorbed lipids must be resynthesized. This is very important stage. We didn't talk about any resynthesis for the monosaccharide or for amino acids. They will go through the membranes of enterocytes, through the membranes of endothelial directly to the blood flow. Here not. So our monoacylglycerols will be attacked by enzyme which is named acyl coenzyme A monoacylglycerol acyltransferase, attacked with the acyls of fatty acids. What is important to say that fatty acids will go to any reactions only after their activation. It does not mean they necessary for the synthesis, doesn't matter, or necessary for the cleavage. Anyway, till this residue of acid must be activated. This very important moment, please put attention. I will repeat again that all of our molecules are participating in the reaction in their so-called active state, activated state. And for different molecules, different activation is necessary. For example, to activate glucose, we told about this many times, we will phosphorylate it. Fructose, galactose, monosaccharides, to be incorporated to the catabolic reaction or synthetic reaction, must be phosphorylated. We compare this with the children, which are very active and very fun, when they will obtain balloons. So these balloons, these balls, air balls, like air balls, we write the phosphate groups for the monosaccharides. Our fatty acids must be activated with the coenzyme A. I shown you the coenzyme A structure when we talked about the activation of residue of acetic acid. Yes, our pyruvate on the second stage was attached with the coenzyme A, big substance from adenine, ribose, phosphate units, pentatonic acid residue, and tier alcohol with the sulfur tier group, which is used to be attached for the residues of acids. It's comparable with the head of kite, to the tail of which we need to glue long tail of fatty acids. So at first, all free fatty acids that are absorbed inside of the enterocyte will be activated with the enzyme which is named fatty acyl coenzyme A synthesase, or simply acyl coenzyme A synthesine. For this we are using coenzyme A, molecule of ATP, which is cut to AMP and pyrophosphate, so nearly energy of 2 microerg bond we are using to activate our acetyl-CoA. It's one of the representatives of acyls, but it is short in chain. Long chain fatty acid, for example, palmitate, oleate, stearate, converted to palmitoyl coenzyme A, stearyl coenzyme A, oleal coenzyme A, and in this active state, by the action of acyltransferase, they are attached to monoacylglycerols. Diacylglycerol is formed, and then triacylglycerol is formed. So one transferase is acting plus one fatty acid, second acyltransferase is acting, and finally we are resynthesizing triacylglycerols. Sorry. But now these triacylglycerols contain fatty acids which are characteristic for human fats, with the special degree of saturation, with the special melting point, human specific. These resynthesized triacylglycerols go for the formation of particles which are named chylomicrons, and then they will go to the lymphatic system. Same cholesterol will be re-esterified with the help of active acyl coenzyme A, and enzyme which is named acyl coenzyme A cholesterol acyltransferase. Fatty acid will be attached to the cholesterol, and in a view of cholesterol ester, also these substances will go to the formation of chylomicrons. Fat-soluble vitamins that also require presence of bile and emulsification together with fats are coming to the structure of the primary chylomicrons, and this particle will be formed. To be transported, doesn't matter by lymph or by blood, these very hydrophobic fats, like triacylglycerols, like esters of cholesterol, which are absolutely hydrophobic, must be surrounded by phospholipids. A layer of phospholipids is necessary, so phospholipids will be also synthesized in the enterocytes or come to them from the lipid which will synthesize them. Phospholipids with a thin layer will surround this very hydrophobic core. And in addition, to increase the solubility of chylomicron, we will use apolipoprotein B48, which is synthesized from amino acid also inside of enterocytes. I shown you the structure of lipoproteins at the previous lecture and told you that all proteins that will specifically synthesize to interact with the lipids in our body will be named apolipoproteins. Why so strange name? At first we need to say that there are several classes of apolipoproteins and they are named with the letters A, B, C, E. Near these letters you can see numbers 1, 2, 3. So this is classes and subclasses of lipoproteins. This depends from their structure, from their primary structure and their properties. Especially this apoprotein that will be attached to chylomicrons belongs to class B. And what is interesting, the same gene for the which is coding the apolipoprotein is expressed in the liver cells, and liver in their order, in its order, will synthesize apolipoprotein B100. It means that in the liver, this protein is synthesized completely with the long chain of amino acids. 100% of the gene will be expressed and 100% of the protein will be synthesized in liver. Enterocytes express this gene not completely, and only 48% of the primary structure of apolipoprotein B100 will be synthesized in enterocytes. And due to this, it is shorter apolipoprotein than liver, but they belong to the one class, and primary structure in them is equivalent to the 48%. Apolipoprotein B48 is characteristic especially for chylomicrons. So after absorption and resynthesis, combination with the apolipoprotein B48, chylomicrons will go through the membranes of enterocytes by exocytosis to lymphatic ducts. Then lymphatic ducts will be collected and go to the venous system. So from the difference in absorption of lipids in comparison with amino acids and monosaccharides, that primarily they are absorbed to the lymph. And if you will look to the transparence of the lymph after eating of fats, you will see that transparency is decreased, and lymph will be same with the milk. It will be so-called oil lymph. Then, when particles of chylomicron will reach blood, our blood serum, if you will see it, will be also less transparent due to the presence of this chylomicrons particle. So, what will be the fate of chylomicrons after their formation and absorption in the intestinal, after digestion of lipids? Through the lymphatic ducts, they will finally come to the blood vessels, and in blood vessels they will meet other lipoprotein particles which already were formed by liver and which contain a little bit another combination of lipids. We will talk very detailable about this synthesis of lipoproteins by liver and their transport and their exchange. At this moment we are talking about only chylomicrons, but in addition to its apoprotein B48, during interaction with other lipoprotein particles, new proteins can appear in the structure of chylomicrons. For example, apoprotein C2, which is synthesized in liver and come to the chylomicrons only in the lumen of the blood vessels during the meetings with other lipoprotein particles. It is possible to change position of apolipoproteins from one particle to another one. So chylomicrons now will be more beautiful, take new apoproteins from the lipoprotein particle formed in liver. They move through the lymphatic system to the capillaries already with the additional apoproteins, and the task is to transport lipids that were digested and absorbed in the gastrointestinal tract to the adipocyte mainly, because we prefer to oxidize glucose in our cells and fats that we are obtaining. Our metabolism is regulated by the way when fats will be used for the storage, not primary to the oxidation, and glucose and other monosaccharides will go to the oxidation, lipids for the storage. So the task of chylomicron is to transport dietary lipids absorbed in the gastrointestinal tract to the adipocyte mainly, or to the muscle cells. So we will not start immediately to cut fats that we are eating. We mainly will put them for the storage. As we told, inside of the chylomicron, we have triacylglycerides and we have esters of cholesterol. So to put these triacylglycerols to the adipocytes, we need to cut them again. And for this, we need enzyme which is named lipoprotein lipase. This lipoprotein lipase will help to cut triacylglycerols and come free fatty acids inside of the adipocytes, where again triacylglycerols will be resynthesized and finally will be stored in our adipose tissue. New lipase, again many lipases names we pronounce today. Lipoprotein lipase, put attention, pancreatic lipase, where it can be localized. If it is synthesized by pancreas, it can be localized only in pancreas or in duodenum and small intestine. Lipoprotein lipase, where it can be localized. Lipoprotein particles we have inside of our blood. So lipoprotein lipase, this is an enzyme not from gastrointestinal tract. It is attached to the endothelial cells. So on the bottom of the blood vessels, we have this enzyme. So, as I told you, after absorption, our lipoprotein particle, look, inside we have cholesterol, esters, triacylglycerides. This is core, hydrophobic core of the chylomicron, surrounded by phospholipids and cholesterol, Look, yes. And finally interacted with the EPA protein B48. This is from enterocytes. And when it is swimming in the gastrointestinal tract from other lipoprotein particles like high-density lipoproteins, epiprotein C2, C3, epiprotein E can come to the chylomicrons and increase their solubility to help them to be transported by their water. Our blood is water solution, so this, how to say, gifts from the high density lipoproteins will increase solubility of chylomicron, and finally it will go to the adipose tissues where fatty acids will go inside of these tissues, and subsequently our lipoprotein particle will lose this additional epiprotein again that will be taken back by more high density lipoprotein particle. And finally, more poor chylomicrons, which are named remnant, will go to the liver, where there are receptors for remnant chylomicrons, and these particles will be taken by liver, and liver will decide in further what to do. About this a little bit later. At this moment, you must understand that chylomicrons are bringing the fatty acids from the dietary lipids to the adipose tissue and muscles, and as a remnant particles go to the liver. A little bit more words about lipoprotein lipase. Right place that this enzyme is necessary to cut triacylglycerols from chylomicrons and put the free fatty acids through the endothelial cells to adipocytes, to small quantity of muscle cells. Look to the mechanisms. On the surface of endothelial cells, we have special receptor for the chylomicrons, which are constructed from the polysaccharide chains attached to the proteins and phospholipids of the membranes of endothelial cells. They are combined with the lipoprotein lipase. This is the protein which will recognize chylomicrons by the presence of epiprotein C2. So very important for chylomicrons to meet other lipoprotein particles in the blood and to take from them this gift, epiprotein C2, because it can be recognized by receptor, by lipoprotein lipase, and finally such chylomicrons by the action of lipoprotein lipase. Part of triacylglycerols from the central core of this chylomicron will be cut by lipoprotein lipase on the surface of endothelial cells to free fatty acids and glycerol that can be absorbed, can be nearly absorbed, can go through the endothelial cells membrane inside of endothelial cells and then go to the adipocytes where the fats will be resynthesized again. So several stages of cleavage and again synthesis of triacylglycerols we have. Due to this, I told you that this process is more complicated, of course, than absorption and digestion of proteins or carbohydrates. What is necessary to tell that fatty acids mainly will go to the adipocytes and muscle tissue, and glycerol will be absorbed to the will not be absorbed directly to the adipocytes and muscle tissue. It is not necessary for them. Free glycerol mainly go by blood to the liver, because in liver we have enzyme which can activate this glycerol and use it for the further synthesis of fats. Part of glycerol that will be formed after cleavage, so greatest part of glycerol that will be formed after cleavage of chylomicron triacylglycerols will go to the liver. About lipoprotein lipase, we need to mention that it is activated by insulin. Please write this thing. Lipoprotein lipase is localized on the endothelial cells, not in the lumen of intestine. It's a big mistake. and lipoprotein lipase is activated by insulin. And this explains why we are not oxidizing immediately fats that we ate. At the same moment, together with dietary lipids, glucose is absorbed. Because at the same moment, during the digestion of the food products, pancreatic amylase cut starch and glycogen that we eat together with fats. We are not eating separately fats, carbohydrates, and proteins. Mainly we are eating mixed food where all of these molecules are present. And after absorption of glucose, pancreatic gland is secreting insulin to the blood. And insulin increases the permeability of the cells for the glucose. Insulin stimulates glycolysis, oxidation of glucose. Insulin stimulates usage of excess of glucose for the synthesis of glycogen. And insulin at the same moment stimulates lipoprotein lipase to take fats from the chylomicrons and put them for the storage. This is why, if you will remember that insulin is activator of lipoprotein lipase, you will understand why we are using glucose mainly for the oxidation and lipids we will store. Please, our next material will be devoted to the mobilization of these stored lipids and their oxidation when it will be necessary. Of course, we will oxidize them also. Sometimes it is necessary. We will not only store them without oxidation. But the mobilization and transport of these lipids differ from the transport of dietary lipids. So please differentiate. When we are talking about dietary lipids, they are transported from the intestine to the adipocytes for storage by chylomicrons. And when it is necessary, they will be mobilized from the adipocytes for further oxidation and then transported by blood free fatty acids with shortened chain as a free and long chain fatty acids in the complex with albumin. So this is another part of lipid metabolism, their catabolism, their usage for oxidation. Don't confuse lipoprotein lipase that is present on the endothelial cells and hormone sensitive lipase which is present inside of the cells which are storing triacylglycerols. Again, new lipase, so many lipases. Today are named new lipase, hormone sensitive lipase. This lipase is present intracellular. Our lipoprotein lipase is also hormone sensitive. It means it's sensitive for the insulin. But when we are talking about hormone sensitive lipase, we are meaning lipase that is present in the cells that are storing lipids, adipocytes. So look to the scheme, how lipids will be taken for the cells if it will be necessary to oxidize them. Normally, we prefer to oxidize glucose. Our triacylglycerols are stored in the adipocytes, but there are situations where we have no possibilities to obtain energy from glucose, for example, starvation or long period when glycogen of liver is not so long source of energy for us. Near 18 hours and glycogen stop to be source because it will be cut nearly completely and gluconeogenesis will switch on. But anyway, during the longest starvation, fatty acids will be sources for energy, sources of energy for us. So we need to take them and to transport them from the adipocytes to the place of their further oxidation. It can be liver, it can be muscles, it can be cardiac muscle, or even if from the fatty acids the ketone bodies will be synthesized, ketone bodies will be sources of energy for brain. So how this hormone-sensitive lipase is named also tisual lipase, to show that it is inside of the tissues, adipose tissue mainly. Tisual lipase or hormone-sensitive lipase is sensitive to glucagon and adrenaline. So please, lipoprotein lipase, endothelial cells sensitive to insulin, because it must cut chylomicron triacylglycerols, which are swim from the intestine and bring in dietary lipids for the storage. Glucagon and adrenaline tisual lipase activators, because they are secreted contra insula. So when there is no glucose for the oxidation. Glucagon and adrenaline, we talk about this system of their influence to the intracellular enzymes. At first, acting to the receptor, it is not necessary to come inside of the cells. The membrane receptor of glucagon and adrenaline is combined with enzyme adenylate cyclase, which convert ATP to cyclic AMP. Cyclic AMP is an allosteric modulator for protein kinases. Protein kinase enzyme which is necessary for phosphorylation of intracellular enzymes. To be active, protein kinase, which is heterotetrameric structure, the regulatory subunits must interact with cyclic AMP. Dissociation of these subunits will reveal catalytic subunits of protein kinase, and active protein kinase will convert lipase, which is tisual or triacylglycerol lipase, to the active state by phosphorylation. The similar mechanism for the activation of glycogen phosphorylase. And maybe it will be convenient to you to remember that adrenaline and glucagon through the protein kinase stimulates degradation of polymers of glycogen and big molecules which are triacylglycerols which we are storing. So this tisual lipase has three names. These are synonyms. You can name it this triacylglycerol lipase, you can name it tisual lipase, and you can name it hormone-sensitive lipase. Indicate that all three names are synonyms. You can use all of them, any of these three names. Tisual lipase, triacylglycerol lipase, hormone-sensitive lipase. And differ from lipoprotein lipase from the gastrointestinal tract, pancreatic, gastric, lingual lipases. Active triacylglycerol lipase, active tisual lipase, adrenaline sensitive lipase, will cut triacylglycerols that we have inside of adipocyte to glycerol and fatty acid. Glycerol, as we told, can go to the blood easily with itself. Short chain fatty acids also with themselves. And long chain fatty acids must be combined with albumin for the transport. This is one of the functions of albumin, to transport fatty acid, put long chain fatty acid. This protein has many hydrophobic sites on the surface, especially for the transport of hydrophobic compounds. One of these compounds, these are fatty acids with long chain. And then in the complex, with the complex of albumin, these fatty acids goes to the oxidation in the tissues. So this is absolutely another transport, another stage of lipid metabolism. Not after digestion, only in case when the glucose can give us energy, fats that are stored in adipocytes will be mobilized from their depot, so-called mobilization of fats from the tisual depot. One more, please note that free fatty acids will go to the cells, here for example to hepatocytes, but they can go also to muscle cells, to cardiac muscle, to other kidney cells. different cells, in the complex with albumin. And inside of the cells, then the activation of them will be, and oxidation in mitochondria, about the transport through the mitochondrial membrane, about activation. This is the next stage of our discussion. So here you must only one more time note that free fatty acids, long-chain fatty acids, are transported to the peripheral cells with the help of albumin. And do not confuse that dietary lipids, dietary triacylglycerols and fatty acids in their content are transported with the chylomicrons from the intestine to the places of storage, not to the places of oxidation. Now, more detailable, let's look to the reaction of activation of fatty acid. We thought that this reaction also was used by enterocytes for the resynthesis of triacylglycerols. For activation of any fatty acids, we need the special enzyme to combine fatty acid with coenzyme A. And due to this, enzyme is named very easy. In biochemistry, everything is very easy. What you want to obtain, to synthesize, and add name synthase, you will obtain name of the enzyme. What we want to obtain? We want to obtain acyl-CoA, active form of fatty acid. So the name of enzyme will be acyl-CoA synthase. The second name is thiokinase, and especially due to this, we are not using the name thiokinase for the citric acid cycle enzyme. Do you remember that it was very strange that enzyme which is cutting succinyl coenzyme A, which is revealing coenzyme A and converted succinyl coenzyme A to succinate and free coenzyme A, we are naming succinyl coenzyme synthase by the opposite direction of the reaction. It's necessary to prevent the confusion with this acyl coenzyme A synthase, which is also named thiokinase. So this enzyme catalyzes conversion of our fatty acid to so-called active form, active fatty acid, or acyl-CoA. One of the example of acyl-CoA we very good know, it's acetyl coenzyme A. So all acetyls coenzyme A are acyls, but not all acyls are acetyls. It's necessary to remember. Of course, this is not easy to activate coenzyme A, and for this we are using ATP. Look to the reaction of activation. Now it is necessary to write and name that this is activation of fatty acid. Here we have fatty acid, it doesn't matter what tail it has. Here we have coenzyme A with tier group, yes, where acid will be attached. And the mechanism of thiokinase reaction is to transfer this tail to the coenzyme A. The substance which we will obtain is named acyl coenzyme A. And as we told, we are using ATP for this. You can see that in this reaction, 2 macroergic bond energy we are using, because as a result we are not obtaining ADP, we are obtaining AMP, monophosphate, and pyrophosphate is cut after this pyrophosphatase will cut this pyrophosphate to 3 phosphate, 2 phosphates. Doesn't matter. What is important that here 2 macroergic bonds are used, and due to this, in most of the foreign textbooks in biochemistry, when energetic effect is calculated, more often for the activation, 2 molecules of ATP subtracted from the final result. Put attention and note and write somewhere that in Russian school, scientific Russian school, we are calculating -1, because all the time we are calculating for the moles of ATP, not to the quantity of macroergic bonds, but to the quantity of molecules of ATP. So when energetic effect we will calculate, we will put -1. here for the activation. Anyway, this activation is necessary for all fatty acids. So in calculation of energetic effect of fatty acid oxidation, all the time we will calculate -1. For glucose, we calculated -2, if you remember, because two reactions of phosphorylation we have in the investment stage of glycolysis. At first, phosphorylation of glucose, then phosphorylation of fructose 6-phosphate to activate glucose -2 ATP. Here, we will calculate -1, not -2 in meaning of quantity of macroenergy gone. Then we need the special substance which is named carnitine to transport this residue of fatty acid inside of mitochondria, because our fatty acids will be oxidized in mitochondria. You know that pyruvate also must be transported to mitochondria because enzymes of citric acid cycle are localized inside of mitochondria, and also a respiratory chain that will take reduced equivalence and transport electrons to the oxygen are situated in the inner membrane of mitochondria. So oxidative, the most important oxidative processes are localized in mitochondria. Membrane of mitochondria is not permeable easily for the substances. You know this very good. Membrane, this is the place where potential is formed, so permeability of the substances is checked and regulated very strongly. So long chain fatty acids pass through the mitochondrial membrane with the connection with special molecule. This is carnitine. This is special substance. Look, you can see here in the structure of carnitine you can find choline, yes, alcoholic group, ethyl, and carboxylic group here to be attached to the acyl of residue of acid. The special enzyme which is named carnitine acyl transferase, look, carnitine acyl transferase. We will put one because we need to explain that this acylcarnitine transferase is localized in cytosol. So this reaction is will be performed in cytosol to create substance that can pass through the membrane. This substance is named acylcarnitine. So acyl-CoA, already activated fatty acid, is interacting with carnitine and attached to the through the alcoholic group, by the ester bond, to our carnitine. We will obtain acylcarnitine, and this is a form in which this hidden fatty acid tail will go through the membrane of mitochondria. We know that if something is prohibited to pass through the border, what is necessary to do? It's necessary to hide it. Same like our shuttle mechanism hided hydrogen to pass it, to bring it inside of mitochondria. Carnitine will hide the residue of fatty acid in its structure, and by this way pass through the membrane of mitochondria. Write please this reaction and indicate that carnitine acyl transferase is the most important enzyme in the regulation of oxidation of fatty acids. Most important. At this moment, we will only say that, tell that inhibitor of this enzyme is malonyl-CoA. What is malonyl? We will know further. This is the initial substance for the synthesis of fatty acids. So synthesis and oxidation of fatty acids is regulated also by the special mechanism where only one process can be done. It's unlogic to do oxidation and synthesis at one time. When we have energy, we will synthesize lipids and store them. When we have no energy, we need to cut fatty acids. And in this case, these two processes must switch on and switch off one another. Especially this connection can be done through the substance which is named malonyl-CoA. Indicate this that malonyl-CoA is inhibitor of acylcarnitine transferase and can stop transport of fatty acids inside of mitochondria. It's a narrow moment of the fatty acid oxidation. They must come inside of mitochondria. If they will come, they will be oxidized. If they will not come and be in the cytosol, they can be used for the synthesis, for the accumulation, for the combination with glycerol, for the synthesis of triacylglycerols and phospholipids. When acylcarnitine will go inside of mitochondria, there is the same enzyme, but with the indication that this is already mitochondrial acylcarnitine transferase, number 2, will do the opposite reaction. Acylcarnitine will react again with coenzyme A, and transferase is changing the position of our acyl from carnitine to coenzyme A. We again will reconstruct our acyl-CoA, active form of this fatty acid, that after this will go to the oxidation. Carnitine, free carnitine, can return to the cytosol and then take another long-chain fatty acid to transport it in the mitochondria. So reactions are the same, but in the cytosol, acyl coenzyme A put acyl to the structure of carnitine. Carnitine is returning this acyl to the coenzyme A inside of mitochondrial matrix and go back to the cytosol. So this is the shuttle system. Look at it. From the cytosol, acylcarnitine transferase 1 take residue of fatty acids from the acyl coenzyme A, put it to the carnitine. Acylcarnitine goes through the membrane. Special transporters are used for this. And inside of matrix, carnitine return this acyl to the coenzyme A. Acyl coenzyme A is reconstructed, so it was here, now it's here. It is here. Coenzyme A is free. Carnitine is free, and carnitine will return back. Note that enzymes are named with the same carnitine acyl transferase, but number 1 indicates that this is cytoplasmatic enzyme. Number 2 indicates that this is enzyme from mitochondrial matrix. So carnitine is a carrier for fatty acid. Start to remember this. After this, the process of oxidation can be started. More detailable, we will discuss it with the names of enzymes, with the mechanisms of reaction next lecture. At this moment, just write that oxidation of fatty acids takes place in mitochondria of many cells and organs, but not in the brain. In the brain, beta oxidation will be not done. Brain will take ketone bodies. About this a little bit further. Oxidation of fatty acids is aerobic process, so it requires the presence of oxygen. Mitochondria, oxygen. Conditions necessary for fatty acid oxidation. About fatty acid oxidation, we will talk next lecture. At this moment, thank you for your attention. We are finishing. Be happy. Be safe. Goodbye.